Welcome back to the lecture series in cell culture technology. So, we are into the second week and today we are starting the second lecture of the second week. So, the thrust area of this lecture will be mostly layout and design of a cell culture facility. So, as you go through the textbooks about different kind of layouts and designs, there are certain salient features which will be highlighted. So, today's lecture I will be talking exclusively from my personal experience of setting up cell culture facilities in different parts of the world under different financial constraints and for different kind of purpose. So, the first and foremost important thing which occurs to me should be taken into account that what level of facility are we looking forward to, what is the objective. I'm not talking about like how big, how small, what is the objective. Say for example, so tell you this, if you are setting a facility in a lab which is working on say cell biology or some like development biology or something, and you wanted to have a small cell culture facility, the requirements will be different as compared to say for example, it's a biosensor lab, you just use the cell as just to maintain some kind of a cell, to put it on some kind of sensor device, cell based biosensors of course I am talking about, then your requirements will be kind of different. Similarly, if it is a tissue engineering lab where cell tissue interaction with the materials are being tested, again the requirements will be of different type. Yet if you are a chemistry department which wants to screen different kind of fluorescent molecule, different kind of anti-cancer drugs or different kind of newly formulated compounds, your requirements will be different. So, you see, or say for example, so let me jot down these points. Okay. So, we are into week 2, lecture 2. So, W2, L2, and so what we are dealing today is layout and designs, layouts and designs of cell culture facility. Okay. So, now I told you that there are different kinds of facilities. Say for example, what are the major kind of objectives. So, one we talked about, if it is a cell biology lab and it wants to have a facility whose major work are cell biology or a development biology lab, and I will come later what are the specifications. Say for example, a lab working on cell based biosensors or a lab working on cell tissue engineering or a chemical biology lab where essentially the idea is screening different drug molecules, their interaction with cells, or not only drug molecule, different say fluorescent molecules. Similarly, cell tissue engineering, the major thrust area is cell tissue material interaction. Okay. Cell-based biosensors is mostly cellular component as biosensor element. Similarly, cell and development biology, if they wanted to have a cell culture facility, okay, so they have something, regular maintenance of cell line. And again, it depends on what kind of development biology you are doing. You could have a in vivo, where you are exclusively dealing with animals and histology, or you have an in vitro development biology, where you are doing everything outside the system with isolated cells. Then definitely cell lines and primary cultures. I'll come later. What does that mean? And of course, in the point of cell biology, there you are doing with different kind of cell lines and all this stuff. So, I mean, this list can go on, what all you can do. Similarly, there is another fifth group which exclusively work on developing cell culture systems itself. These are the labs which exclusively work on medium development, medium for cell culture, of course. They work on substrate development, substrate for culturing the cells. These are all for the, okay. Then they do a different kind of, or let us put it like this, there is another emerging area where there is an area of biomems, cell based biomems structure. MEMS here stands for micro, electro, mechanical, S stand for systems. Okay. So, there are cell based biomems structures which are being developed for, say, for example, history goes back to like things like Campanot chambers. We will come later what are those different aspects are, something like modified, one second, Campanot chambers and several other things, people work on biorobotics and all those areas. So, first and foremost thing what has to be understood is what is the objective, because there are constraints. What are the constraints what one comes across? So, first, what is the objective and what is the purpose? So, let us get all the functions right. What are the functions, what are important to understand? So, the major is objectives or the purpose / purpose. So, we have already talked about the purpose in the previous slide. You see, these are the different kind of purpose we are telling. So, you should know what is your purpose, what kind of lab you really wanted it to be. After the purpose comes the constraints. What are the constraints? The first constraint which you will see all over the world, a common constraint, irrespective of the fact whether you have money, whether you don't have money, how much money, is space. Space is a huge constraint all over the world. Like if you go to places like National Institute of Health at Maryland, Bethesda, you'll be surprised, like, you have seen the office cubicles. Most of you have seen sometime or other, small office cubicles. It is almost, that is the size of the facility you get for cell culture. And there are people who will be crammed in there, like, 3 or 4 groups sharing such facilities. So, all over the world, this is very critical because getting a space, getting designated space with all the, like, the vacuum systems and all those things, it's not easy because you need certain special things if you want to make it more and more sophisticated. So, space is a big constraint, and the next constraint comes, if your space is a constraint, related to it is space management, and that is where the whole design layout comes very handy. Space management. This is very, very critical because first of all you are constrained by space. So, let me put it like that, space constraint, and then you have to manage the space. And if you talk about the manage the space, then we have to talk about what will be the kind of equipments or the size of the equipments you wanted to get for such constrained space. Point 2, we will again come back to this space constraint. Point 3 is your financial constraint, because wherever you set up a lab, you will always have a financial constraints and optimization. So, this arrow, now I am again redoing that arrow, a space and a finance, these has to be optimized very neatly, and then comes based on your objectives. What are the objectives you have set for yourself? You have to decide what level of sophistication you wish to achieve. What does that mean? Say for example, your lab regularly maintains a particular cell line. I have already talked to you about cell line, right, in the first week. And you just, your job only is to maintain the cell line, and you just passage them every week after week, subculture them and grow them, maintain them, and you just take them and do your experiment. You probably may not need a very high end microscope for that purpose. I mean, you just have to observe the cells, they are doing fine. If that is your objective, then you are not going to spend on a very high end microscope, right. On the other hand, if your lab does a lot of cell biology work and that completely relies on your cell culture, then probably you may need a very high end microscope facility, and you may prefer to have a microscopic facility then and there adjacent to your working bench. So, these decisions are very critical, provided you are very sure what is the purpose and what is the objective. And adding to the purpose and the objective is, there is another thing which comes, is plan for expansion in next 10 years. Because unless you have a very clear cut plan, this is how you want to explore. Or say for example, you are not very sure. Suppose you are a new faculty starting something, you are not very sure. Yeah, I may do this. I mean, that happens with all of us, right? You know, when I set up my lab, I still remember. And as a matter of fact, when I set up labs for my employers, I always told them this simple thing. Like at this point, I do not know. Like I may love to, explore this area. So what do? You do not consume the whole area. You kind of cover the area by some way or other. It should not look like that you are not utilizing that area, but you should have a buffer space. So tomorrow, say 5 years down the line, you wanted to explore that site, and I wanted to do this, but where I am having space because I have got all this big giant stuff and I have covered up everything. That should not happen. That farsightedness is very, very critical, that we all grow. Our desire changes, our scientific problem demands us to explore areas. How you do it? So that brings you to that critical point. What I was mentioning earlier is both. This is a direct function of your space management, okay, as well as your, at that point, financial management, okay. These two are very critical, and I used another one word. I am just, before you forget and I forget, maintaining, and this is, I believe, each one of us should, keep in mind, maintaining buffer space for future expansion. This is very critical. So unless you have that buffer space, you will kind of, you'll realize that like, I should have, saved that space. I could have utilized it later. Okay, that comes the next. What level of sophistication you wanted to achieve, and what are your plans for expansion. Next thing, if I put this as E, next important thing is that what is the level of biosafety are we talking about? Say, and this of course comes in the objective in the beginning, level of, once again, biological safety. What does that mean? So before I get into that, so this is of course a very, very direct aspect. So say for example, I wanted to work with a very deadly virus called Ebola, which is a bioterrorist agent, or I wanted to work with Marburg virus. Hell, I mean, this is like something, if I am not wrong, there are a handful of labs. Maybe across the world there will be 3 or 4 labs who can handle that level. Or you want to work with a very deadly strain of TB, tuberculosis, which is common in India. Are we equipped to do so? Because the person, the user, suppose I am using this. So first of all, I am putting myself into a threat. I may, get the infection. The other person who are working, they may get an infection. And not only that, and if we can't contain it, then the whole area, the whole community will get an infection. and these are deadly stuff. So, first and foremost, as the part of the objective, when I talk about the purpose, if you remember the purpose out here, what are you working on? So, now I add few more points. Are you working on, say, now I am adding few other points out here. Say 1A is this, then are you working on infectious biology? Are you working on virology? Infectious biology, virology. So, that brings us to the point of level of biosafety. The contrary, if you are using a kind of regular cell lines, it really doesn't matter. I mean, nobody really bothers. I mean, it's perfectly fine. Or you're using some kind of, some straightforward materials directly derived from biological system, should not be a problem. Then comes another interesting aspect. Since we are living in an era of, so adapt to that. In cell biology section and development biology section, are we working on transgenics? Does it involves DNA transfer? Are we working on knockouts? Are you working on RNAi? So, that demands different level of precautions which has to be taken, because the very moment you are handling with the DNA material or any kind of genetic material, there are certain rules and regulation or the law of the land. Wherever you go, whether you are in India, whether you are abroad, any country, every country has very, very stringent rules, extremely stringent. So, one has to know those rules and regulations a priori or much beforehand before you design. So, I want to do this, and then you realize that in that particular building, you really do not have the proper kind of exhaust, or you do not have a proper way to contain the building, you do not have a proper runway or escape routes, and you wanted to open a facility which kind of put others in hazard. So, these are very critical, exceptionally critical, and you have to be very, very clear that what are my, because you are dealing with systems which not only is hazardous to you, could be hazardous to others. Unless you are sure, unless you are very clear in your mind, you should not lay down. First of all, think over it. That is why I am jotting down this point, what is the objective? And these are the points which textbooks will not say, but when you will go to the day to day life, if you take any textbook, I mean, they will kind of brush aside these things, but the practical reality is this. These are, you come to know once you go to the job, once you have to set up a lab, then also, oh no, you cannot do this, oh no, this is not allowed, oh in this building we cannot do this. And I completely understand their point, because certain buildings are not even made for that purpose, as a matter of fact. So, these things you should keep very, very clear in your mind what level, because based on that we have these levels called Biosafety Level 1, Biosafety Level 2, Biosafety Level 3, Biosafety Level 4. And for those who are little inquisitive to learn about much of these dangers, should read a very nice book, The Hot Zone. If you find it online, or I'll try to if someone of you can't contact me, if I have a e-copy, I'll forward this. Okay, you should read this book. This is a very interesting book about deadly viruses escaping out and how that could influence our life big way. Okay. So, next is this biosafety level, what you have to be very, very critical. And in that, of course, I have already mentioned, you have to be very clear, are you working with DNA, are you working with other forms of nucleic acids and what so on and so ever. Then you have to realize in that objective line, am I going to use live animals? In other words, say for example, I wanted to work on primary cultures. So, primary cultures are essentially when for culturing you directly derive the tissue from live animal. You have to kill the animal, or if the tissue you just take a small part of the skin, then you can give the animal necessary first aid and anesthetize the animal, take the tissue and give the animal necessary first aid and okay, good to go. Or you want to sacrifice the animal. Now, if you want to sacrifice the animal and you want to pull the tissue out of that animal, that demands different kind of facility support. Because when we are talking about sacrificing the animal, what we are essentially talking about, you have to have animal sacrificing facility, right? Not only that, the biological waste which are coming out, first of all, any animal killing has to be done in a humane manner, in an ethical manner. It is one underlying prerequisite. So, first how you are killing the animal will come in depth. There are different ways to do so. Okay. Then how many you wanted to kill. Then once you take out your desired tissue, how you dispose the animal, where you bury it, or where you put it to rest. These are very ethical and very human aspects, which one has to be very clear that how really you want to do it, because then there are every institute across the world, recognized institutes have animal ethics committees. Then you have to take ethical clearances. You should know what all ethical clearances you needed to know. Barring aside some animals, like you can work with fishes without any ethical clearances, and there are few lower animals you can work with, possibly Drosophila and all those small systems where you do not need ethical clearances. But apart from it, the very moment you move to the rodents, guinea pigs, they all need ethical clearances. So, what sort of ethical clearances you will be needing? So, whenever we are talking about live animal or primary culture from live animal, so all these points will come to play, that sacrificing method, sacrificing route, carcass or the cadaver disposal, and most important, they all fall under, do you have to have an ethical clearance. So, you realize even much before I hit upon the topic of how the layout of a lab will be, there are other things which one has to take into account. Okay. So, I will close in here. We will continue, we will resume our journey of understanding what all basic prerequisites one has to take before one think of developing one of the state of the art facility. So, ponder upon it, think over it, and thanks for your patient listening.